Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another really fascinating guest today involved in creating a better tomorrow via his work. Today we have the honor of being joined by Professor Dr. Gordan Lauc, who is Professor of Biochemistry and Molecular Biology, University of Zagreb, Faculty of Pharmacy and Biochemistry, as well as the founder and CEO of Genos, a research-intensive small enterprise located in Zagreb, Croatia, with a core expertise in both molecular genetics and glycomics, which performs contract research, analysis, services for numerous universities, hospitals, private individuals across Europe and overseas. Professor Lauc also is the Chief Science Officer of GlycanAge, which is a company that has groundbreaking tests that's involved in analyzing personal glycomes, glycobiomes, for insights in both health, wellness, and monitoring biologic age. Professor Lauc graduated with a degree in molecular biology at the University of Zagreb. He did his Ph.D. in biochemistry there as well, got his postdoc training in both the Institute for Medical Physics and Biophysics at Münster and at Johns Hopkins down the road here in Baltimore. Since 1993, he's been on the faculty of pharmacy and biochemistry. And between 1998 and 2010, he's also been part-time University Osijek School of Medicine, where he founded a DNA laboratory for the ID of war victims. He also served as a vice dean for science between 2001 and 2005. Extensively published. He's also the chair of numerous conferences, including the European Science Foundation Exploratory Workshop on Glycoscience, as well as president-elect of the International Glycoscience Organization and member of the steering committee for the European Glycoscience Forum. A lot of important things to get into with us today. Professor Gordan Lauc, welcome to our show.
Thank you for the invitation. You did your homework.
You know, it's a fascinating area, needless to say. You know, I've watched many of your presentations where, you know, you've described the human glycobiome as, while the most complex area of biology, clearly one of the most neglected. And then when I dove into sort of your several hundred publications there in the peer-reviewed literature, you were involved in this back in the early 1990s. Talk a little bit about how you chose the most complex area of human biochemistry as something to jump into at the very beginning of your career.
Yeah, so it's actually an interesting story. So I was a first-year undergraduate student, very ambitious. I was always very ambitious. So I tried to persuade a local professor of biochemistry to let me work in his lab. And normally, you know, even I now don't accept the first-year undergraduate students because they don't know anything. You don't want them in a lab. So the guy told me, you know, okay, if you want to work in my lab, you have to bring me a brain of a calf, but in less than 4 hours after the calf was slaughtered. Oh gosh, that's difficult. How do you get the brain within 4 hours to the lab? But I managed somehow, so I got the brain, fresh brain to the lab. And then he told me, okay, that was good, but now you have to homogenize the brain, meaning you take a small glass tube and a small glass piston, and then you just make it into the one mesh. And this took 3 days, 8 hours per day. So after I spent 3 days, 8 hours per day, just homogenizing that calf brain, he said, okay, now you can work in the lab. And the lab was a glycobiology lab. So this is where I did my first glyco experiments. I think that the first lesson there is, you know, being smart is not enough. You have to work hard, and this is mostly how I also now choose my team members. In addition to being smart, they have to be willing to work hard.
If I could quote for a second your editorial entitled "Sweet Secret of Multicellular Life." Carbohydrates have been at the center of human interest since the very beginning of civilization, but we've only begun recently to understand the importance of complex oligosaccharides attached to protein or the backbones. This is probably not surprising, since branch structures of sugars make analysis of glycoconjugates significantly more challenging than either the analysis of linear DNA or protein sequences. And yeah, I mean, in my own experience in this space, going back a few decades myself, people always talked about the infinite possibilities of the proteome. If you add the glycome on top of that, you're talking multiple, infinites here. Could you please take us just on a little walk through generally what we refer to as the human glycome? We hear about glycosylation of our proteins and the importance for function. We see glycosylation of natural products, and we'll get to digoxin in a little bit. Talk about glycome in general, how it's everywhere in sort of the human system.
So if we think about biology, all the way back to the beginning of the life, initially life was RNA-based. So we had only RNA, which was performing both the function and holding the information. And then sometimes during the evolution, RNA kind of invented DNA to store the information in a more reliable way, because it's double-stranded, it has thymidine instead of uracil, so it's more stable. And invented proteins to make a more variable structure, because RNA is not very variable molecule. It's only 4 nucleotides, it's four bases. It's very hard to make different structures. So the proteins were invented. So the first evolution and evolution was nucleic acids. The second one were proteins. And then when we think about life becoming more complicated, so bacteria have approximately 5,000 genes. Humans and similar organisms have 20,000 genes. And we are not only 4 times more complex than the bacteria. We are probably millions of times more complex. So to enable all this complexity, life had to find a way how to make a more complex structures than only proteins. Because each protein is defined by a gene. You have to have a sequence of nucleotides to make a protein. So there is alternative splicing, you can make some changes, but more or less protein is defined by its gene. And to change a protein, you have to change a gene. And you change a gene by random mutation. So not really, these things are moving slowly. And then life invented glycans. Glycans are chemical structures which are being attached to proteins. So they're chemical part of protein. They have a covalent link to proteins. But the structure of a glycan is not encoded in a gene. It's encoded in a network of dozens or hundreds of genes. So there is another layer of complexity. So not only the sequence of letters in the DNA which defines a protein, but interaction of multiple genes and their epigenetic regulation and the environment, which make a decision, how will the glycan look like at the end? So it's another layer of complexity which enabled us to become much more diverse. So each our protein can now be multiplied in hundreds or thousands of different glycoforms. So the simplest glycoproteins like immunoglobulins, which have only 1 glycosylation site, so 2 chains, so 2 sites, will split a single polypeptide sequence into hundreds of glycoproteins. So if we talk about complexity of life, genes, human has around 20,000 genes, proteins, maybe 100,000, maybe 200,000, but that's the order of magnitude. Glycoproteins, 10, 100, or even more million different structures. So we should think about glycans as the ultimate layer of complexity, which is fine-tuning our biological functions. So analogy I like to make is with cars. So we can have the very basic car, the cheapest version you can buy. More or less, you have a wheel, you have seats, and then you have an engine, and it moves. And then you can have the fanciest racing car. And this is the difference between a protein and a glycoprotein. So the basic protein can perform some functions. Glycoprotein is actually what makes life possible. Another analogy I like to make is with birds. Birds are fascinating animals. They survived when actually birds are dinosaurs, but evolved. So if we imagine a bird without a single feather, it's still a bird.
Right.
It can eat, it can walk, it can see, but it cannot fly. And unfortunately today, many scientists are doing research on this type of a model. So they take a bird without the feathers, take a protein without the glycans, and then study the function of a protein.
Mm-hmm.
You can do some good science, high quality science, but you will never understand the full function of a protein. So this is why we are still failing to understand big part of biology,
mm-hmm.
because many scientists are still ignoring protein glycosylation.
And per your 2015 paper, where you really do a major synopsis of the human and glycome mechanisms of disease, and you point out here that there's been technological development to, I would say, get rid of the fear, let's say, that has existed for a long time. Maybe why people haven't looked at those feathers. Obviously, we know a lot about genomics and proteomics and these tools. What are some of the tools that you've seen as, the last couple of decades you've been involved in this space? Talk about the tools that have evolved that have allowed you, well, you've always been, you've not been afraid of it, but a lot of people have been. Talk a little bit about some of the tools that have evolved alongside you, make things slightly easier when you're looking at these extremely complex glycome structures.
Okay, so I'm terrified of analyzing glycans. We are still only scratching the surface. We are still, we can look, for example, today at the composition of the total glycome. So take a tissue or take a protein, cut off all the glycans and look at the ratios of different structures. For individual proteins, maybe 10 of them, maybe for 10 proteins like alpha-1 glycoprotein, C3 complement, IgG, IgA, IgM, there are methods to look at the glycosylation.
Yeah.
For majority of proteins, we still cannot do it. For majority of membrane proteins, we still don't have methods how to properly study their glycosylation. Our sensitivity is still not good enough to go at a single cell level, and let alone the single protein in a single cell. So we are, when we compare to the genomics, we are in the 90s of the last century. Yeah. So we can do some things, but most of the things are still not working. But what can be done today? One thing is, yes, we can look at the glycome of a tissue or a plasma or whatever fluid, and we can get a reliable composition of different glycan structures in a large number of people. So for example, my lab already studied close to 200,000 people. Mm-hmm. So we can look how glycans change in the disease, with different medicines and so on. Yeah. For some proteins, we can fish them out, like immunoglobulins or alpha-1 glycoprotein C3. And then when we fish them out, we can look at their glycosylation at the glycopeptide level. So what does it mean at the glycopeptide level? So we take the whole protein, we cut it in pieces, and then for each individual peptide which we generate, we look at its glycosylation, because a protein can have a multiple glycosylation sites. So if you take all glycan, if you remove all glycans, then you lose the information about where is this glycan coming from on individual protein. And yes, we can quantify them today. We still don't have tools to change them in a living system, but this is also changing. Actually, the Nobel Prize for Chemistry this year was awarded to Caroline Bertozzi for developing a method to modify and study these glycans in a living cell. So definitely methods are being developed. We can do more and more today, but just think about how much the field of genomics have changed in a decade.
Yep.
From thinking about the individual genome being nearly impossible to now it's commercially available for $100, $200. You can get all letters in your genome and reasonably quickly. Or with the Oxford Nanopore, you go in the middle of nowhere and sequence something you collect in the nature. So I think within the next few decades, we'll see a similar change in the glycomics. So glycomics will become widespreadly used. It will enter the clinics. And I think very soon it will be studied much more than the genes or the proteins.
You mentioned IgG, immunoglobulin G, and you've published extensively on glycosylation in this area, in both aging, disease, homeostasis, the ability to, minor changes to the glycome affecting either a pro or an anti-inflammatory type phenotype. Talk a little bit about IgG, if you would, and why this has been such an important, and we'll get to GlycanAge in a bit, but talk a little bit just about IgG and why this has been really core to your work.
So immunoglobulins are one of the main weapons of the immune system in most animals. Different type of immunoglobulins exist in majority of animals. But here there is one imperfection, and that is that all this recombination of the segments of immunoglobulins which generate these so-called hypervariable regions, which recognize the foreign molecules, happen very early in life. So you define your repertoire of B cells relatively early, and then you can only fine-tune it with a somatic hypermutation. So you generate a sequence which will eventually recognize an antigen. But antigen can be a virus, can be bacteria, can be a food we eat, or something we inhaled. So our immune system has to make a decision what to do with that antigen later. So after the protein sequence is already defined and the gene is more or less formed, the decision what to do with that molecule has to be made later. And this is where glycosylation comes in the game. So antibodies have two domains. One domain is so-called FAB, or fragment antigen binding. It binds to an antigen with kind of two hands. And then there is a constant domain called FC, or fragment crystallizable, it's a historical name, which then decides to which effector mechanisms will this antibody lead. Effector mechanisms are other proteins, other cells, which will bind to that antibody and then either activate a mechanism to kill a bacteria or activate a mechanism to kill a virus-infected cell or decide to ignore that antigen. And often we see people who have antibodies which can be disease-causing, which can be allergy-causing, but people have no symptoms. They have no symptoms because these antibodies are silenced by a type of glycosylation which inactivates these effector functions. Mm-hmm. So glycosylation of antibodies is a sophisticated mechanism which fine-tunes antibodies for their function. And this is something what pharma industry have realized slightly over a decade ago. You know that there are many of those so-called smart drugs, so wonder drugs, mm-hmm. molecules which solve the problems, and mostly they're antibodies. So you develop antibody for a specific target, and indeed, the good antibody can actually eliminate a disease. You can kill a tumor and all the metastases. Yeah. But most of antibodies today on the market have wrong glycosylation. So they don't have an optimized glycosylation. And recently, the whole new series of so-called glycoengineered antibodies are coming to the market. So you have this more or less the same antibody, recognizing the same target, but which is 100 or even more than 100-fold more effective because it has the proper glycan. Yep. So glycosylation, both in the pharma industry, so making better monoclonal drugs, or in our own biology, is essential for regulating immunoglobulin function.
And while we're on the topic of the immune system, say a few words, because during COVID, you published some papers on the glycocalyx layer of our cell membranes, which, we just talked about adaptive immunity there. Here we have a very interesting part of our innate immune system that has protective properties that you published on as well. So just say a few words about that as well, in terms of sort of this other layer of immune response where the glycome comes in.
This is actually the first level of our immunity.
Yep.
So we all know what is the lipid bilayer. The lipid bilayer is a kind of membrane which covers the cells. So
Yep.
This is what we learned about in school.
Sure.
But when we look at the microscope, lipid bilayer is a tiny little lane which is covered with a 50 to 100-fold thicker layer of glycans, the glycocalyx. And this is the real functional barrier between the cell and the environment. And this barrier is particularly important on our mucosas. So of a respiratory tract, of a digestive tract, it's protected with this thin layer of glycans called mucins. And there are very nice videos which can be found on the internet showing how movement of this mucin is eliminating pathogens from the surface of our respiratory tract. So our nose is a fantastic bioactive, chemically active mask, which is trapping the pathogens, both biologically and chemically, and then removing them from our respiratory tract. But a couple hundred years ago, humans started to heat their homes. So evolution didn't have time to react to heating. And a problem with heating is that the relative humidity of heated air is very low, because the amount of water which can be dissolved in the air is correlated with the temperature. So if the easiest way to see that is, for example, if during spring and autumn, when the nights are cold and we go to our car, it will be covered with water. Why is it covered with water? Because the air heats much faster than the surfaces, and the warm air contains much more water than the cold air. So the air coming close to our car will cool down, and the water will get out of the air. It will condensate and form little droplets. And the opposite is true when we heat the air. So even if it is raining, it can be 100% humidity outside. But if it is cold, 0, 1 degree, and we get that air into our room and heat it to 22, 23, 24 degrees of Celsius, the relative humidity will fall to 10, 15%. This is very dry air. And then when we inhale the air is taking water from our nose, from our throat, from our lungs, and it's drying them out. So we all know that feeling if we sleep in some kind of highly air-conditioned hotels, we don't have a good humidification. You wake up completely dry nose, and this is destroying your mucosal barrier, which is removing the pathogens. And this is one of the key reasons why the respiratory pathogens are seasonal. We see waves of infection, usually when the temperature changes quickly, because our body can adapt to low humidity. People live in the desert, but it takes time. You need enough water, and then your mucosal barriers adapt to low air humidity. So when there is a rapid change in air humidity, meaning it becomes colder, I turn on heating, humidity in my bedroom goes down, this dries out my mucosa. And this is one of the key reasons why we have all these pandemics. And actually, I was heavily promoting it at the beginning of the COVID pandemics. Just humidify the air.
Yeah.
No, just evaporate the water. Be careful to breathe the humidified air, and then there will be much less respiratory infection. Because if you go to places like the tropical areas, they don't have influenza and other respiratory diseases. You go there for a week and you enjoy how you don't have any respiratory problems there, just because humidity. We don't think about humidity.
Fascinating, really fascinating. Gordan, one other place I wanted to go before we get into some of the commercial initiatives and so forth. A couple months ago, you published a paper called titled "The Dynamic Brain and Glycome," and here you introduce the reader to the neuroglycome. And if we're not getting as complex, here we have now the incredibly complex nature of the human brain, a barrier system, and then of course all the biology, as you were just mentioning, that we think about, and we're adding the glycome onto this. Not just in disease and aging here, but the very early days of us in our mother's wombs. That glycome, very important for putting that brain together in the first place, holding it together. Talk a little bit about obviously these early days of the neuroglycome, but say a few words about your work here, because I think this is fascinating.
Yeah, so this is extremely terrifying part of our work, because the brain itself is terrifying, and then you add the glycans to that, it's even more terrifying. So I think glycans are important in all biological processes which evolved after the appearance of multicellular life, and definitely brain is one of them. And alternative glycosylation is something which can regulate many processes. And I think this was not mentioned in a paper, but one of the first demonstrations of alternative glycosylation was on a protein called Notch, where extension of the so-called O-few codes is actually activating or deactivating the protein. So we are used to think about phosphorylation as the way to activate or deactivate the protein, but this is just 0-1. You either phosphorylate or not. For glycosylation, you have something like tuning of activity. So by putting different structures, something can be more or less active. And this is very important both during brain development, but also during normal brain function, during the memory formation, and so on. But we know close to nothing about it. So what we are trying to do at the moment, and we haven't even actually published these papers yet, we are trying to map which structures are present, how does they change during development, during evolution, and in different parts of the brain. And there are some fascinating things, like, for example, and this is still unpublished, if you look at the older brain regions, so parts of the brain which evolved a long time ago, they will have mostly glycosylation, which is evolutionary older. So something which would be more similar in a single cell eukaryote than in a multicellular eukaryotes. So as evolution was progressing, as the brain was developing, it was acquiring more modern glycans on an evolutionary scale. So it's that will be a huge area for research, but it will require development of new methods. So with the current methods, we really cannot do much, but with time, we could actually tackle these questions and try to understand how and why is this really important.
And apropos of that, you are a co-director, as well as I was on the steering committee of the Human Glycome Project. Talk a little bit about sort of the ecosystem of folks that you're putting together here. Clearly, this is, as you were saying, this is sort of like the 1990s and really ramping up because there's a lot of work to be done here. But talk a little bit about what has been involved in assembling some of these folks that are less terrified than than the rest of the world in really pushing the glycome forward in terms of our understanding.
So the Human Glycome Project is kind of copying the Human Genome Project. Just the challenge is much larger. And I like to compare it, if a Human Genome Project was putting human on the moon, Human Glycome project is putting human on Mars.
Mm-hmm.
It's way more challenging. But we try to work together to support each other and to approach the instrument providers, reagent providers together and try to persuade them to develop tools which we need.
Mm-hmm.
Because for somebody to develop an instrument, they need to see there is a demand for the instrument. For somebody to start developing good kits, there has to be people who are buying kits and who are developing kits. So we are trying to coordinate this activity on a global level. So all continents, everybody is included. And actually, we try to keep regular meetings of this Human Glycome project meetings. And I think it's making good progress because we are raising the awareness of the importance, especially with the key decision makers. Public comes the last because this is too complicated for general public to understand. But we are talking to governments, to different decision-making bodies, to editors of some journals, and trying to explain to them that glycomics is the next big thing. And then by working together, we can really understand biology much better. And it is working. It's making progress. It's extremely challenging, and there's a lot of work to be done. But I think I'm in the field for 30 years, and maybe for the last decade, I really see things moving faster. And I think it will keep accelerating. So this field will explode within the next decade, definitely.
Talk a little bit, if you would, about GlycanAge, because this is initially connected to what you were talking about in terms of the IgG function changes, pro-inflammatory, anti-inflammatory properties, and so forth. I've seen a lot of presentations with your daughter. You're sort of leading the charge here. Talk just a little bit about what GlycanAge tells us today about health, wellness, aging, and then clearly, based on everything we're talking about, you clearly could have 1,000 products that potentially come out of this in all the different ways the glycome goes. Say a few words about what's going on here.
So initially, this was a problem for us because we were looking to people with diseases and healthy controls and comparing them.
Right.
And then we noticed that if we don't match the controls in patients perfectly for age, we always see a difference. So people usually, patients are a little bit older than your controls, you collect controls in a different way. And whenever we had the patients and controls that were not of perfectly matched age, we saw the same type of a difference. And then when we did our first large population cohort, meaning several thousand people, then we realized glycans change a lot with age. And this was 2013 when we initially published it. And at that time, we had an idea because my lab is also a forensic genetic lab.
Yep.
So we were also thinking maybe we could use this model of age from the drop of blood to determine what could be the age of a person who left a trace on a crime scene. So, you find a drop of blood. Is this some kind of kid who was doing some naughty things and break something, or is there a professional criminal who was breaking in a specific location? So we developed this model of predicting chronological age from glycans. And this was before the methylation age was developed. In the meantime, methylation age clock was developed, and it is more accurate for chronological age than glycans, much more accurate. Glycans were guessing chronological age plus minus 9 years. Then we learned that the difference between the glycan age and the chronological age is best explained with a lifestyle.
Okay.
So people living unhealthy lifestyle would have a higher glycan age than people living healthy lifestyle. And then we literally published over 100 papers showing that the way we eat, our physical activity, even our psychological well-being, different diseases, they all make glycans look older than your chronological age. while healthy lifestyle, moderate physical activity will make your glycans look younger than your chronological age. And then a couple of years ago, we developed this GlycanAge as a product, which we believe can be a very good personal navigator through what we are doing, because we know glycans respond to our lifestyle. For example, if I gain weight, my glycans go up, become older. If I lose weight, they become younger. Because, you know, majority of us more or less know what is healthy and what is not. Yeah. No, there are healthy habits, there are unhealthy habits. But we do not live healthy. We don't change our unhealthy habits, primarily because, you know, if I'm doing something unhealthy, you know, the bill that I will have to pay for that will come in 2 or 3 decades. So, you know, I can do something today and then I will fix it tomorrow or day after tomorrow or a year after tomorrow. But if there would be faster feedback, if I can measure effects of what I'm doing in a more real-time scale, and when we talk about glycans, it's a couple of months. It's not on a daily basis, it's a long-term measure. But if I change something with my lifestyle today, in a few months, I will see what is going on with my glycans. And GlycanAge, I think they have over 15,000 customers so far. And we see that motivation of people is really improving. So they're trying to live healthier. And there is another aspect that all these healthy recommendations, meaning what is healthy and what is not healthy, is based on a standard human. So we take all people, look what they do, and then do statistics and say this is good, this is bad. But there is no standard human. Standard human is imaginative concept. Each of us is different, and we all respond in a different way to food, to exercise, to stress, to whatever. And we did a cool study. It was published only a couple of weeks ago. We had 1,000 people all around Europe on a low-calorie diet for 2 months. Yep. This was really inhumane. I think they put them on 800 calories a day. That's crazy. So they lost an average 10 kilos in 2 months, and their glycans improved. But nobody can live like that. That's right. A torture. That's not life. So they put them on 5 different maintenance weights. So they decided these are 5 different diets, try whether it works for you. So they were hoping to find the best diet. And what we learned is that each of the diets were good for some people, they were not good for other people. So there is no universally good diet. So one has to find a diet which is perfect for him or for her. And this is also where glycans can help. So you try one type of a diet, see the effect. If there's no effect, you try another type of a diet. There's also extreme important interaction between the microbiome and the glycome and a diet, because a diet is also by large determined by our microbiome, and the microbiome is determined by diet. So this 3-edge interaction is what actually makes us healthy or unhealthy. And with glycans, you can actually monitor that. And currently we have a couple of projects globally where we try to use glycans as a readout for microbiome intervention. So you try to fix your microbiome. Let's see how your glycans will respond to that.
While we're on that topic, and you mentioned the torturous diet, but I was wondering, Gordan, could you just say a few words about your paper on stressin and the professional soldier study you did from a couple years ago?
This is nearly 3 decades ago.
It's a cool paper, though.
Yeah. So it's so the methods we had then were really primitive. So it's very hard to interpret what did we measure then using today's standard. But this was during the war in 1990s we had in Croatia. Yeah. And I was then just finishing my studies, and it was extremely difficult to focus on science when there is war around. because we all need to feel that our work has some kind of a meaning and importance. So then I switched to studying effects of stress, because war is the ultimate stressor, and this is it's often forgotten how detrimental psychological stress can be for health. And then we were just profiling the plasma of people who were actually exchanged from a concentration camp. So they were imprisoned after one major battle. They were put in a horrible torture. It was a really bad place. Yeah. And then they were released after a couple of months. And we looked at changes. And then we saw one drastic change with the protein we called stressin. We never found it again because we never had such a horrible cohort where people were really so much tortured. But with time, we did a more sophisticated analysis of people under stress, even animals under stress. And indeed, what we are thinking will have effects on our molecular composition, which at that time, and we are talking about the 90s, was considered to be heresy, because hardcore biochemists would not accept that thinking something will change your molecules. Yep. But even thinking are molecules.
Yep.
There are molecules signaling, there are hormones being produced. So it's not unimaginable. And now we have a lot of evidence showing that what you think is really strongly affecting your immune system. And there is a basic biology behind that. It's the so-called fight or flight response, which exists in all mammals. And the idea is when you are in danger, and we have to think I'm a small rodent and there is a big cat or something like that. So I could either fight or run away. And there is a whole set of biological mechanisms which get activated in that situation, which are improving my probability to survive. There are changes in blood clotting, changes in the blood pressure, changes in circulation. A lot of energy goes to the muscles, to the sensory system. But this energy has to be taken away from somewhere. And they're taken away from the systems which are not needed in the next 5 minutes, when it is decided whether I will survive or I will be eaten. And these systems are digestive tract, reproductive tract, and immune system. Mm-hmm. So the psychological stress, the fear of being eaten, shut down the immune system, reproductive system, and the digestive system. And this is okay. For 5 minutes, we really don't need them. But the problem with the human society is that we are under stress not because somebody wants to eat us, but because I'm late, I don't like my boss doesn't like me, my salary is not high enough, I don't know, whatever. And we continuously activate this stress response. Mm-hmm. We shut down our immune system, reproductive system, digestive system. And if you look what is happening now after 2.5 years of pandemics, we have way more people dying of different diseases. Yeah. We have a huge decrease in the number of babies being born. So all these consequences of stress are now being visible on a population level. And this is something we also saw after the war we had in Croatia.
Gordan, I want to, one other place I want to go with you, and I know we're coming up against a stop for you at the top of the hour, but I really like to ask about, and once again, I apologize going back a couple of decades, but you published this paper on digoxin derivatives as tools for glycobiology back in 2003. I'm a pharmacist by undergraduate training. I spent most of my career in the pharmaceutical industry, which I like to point out was, built on the backbone of natural products. And obviously, digitalis and floxylin, all that goes back to the over 100 years now, to the cardiac glycosides and that whole area of pharmacognosy. And again, just like we're talking about, the fear of glycosylation and all this stuff. A lot of natural products, from what I remember, sort of get left on the lab bench because of some of these unique glycosylation patterns. It could be great drugs, but we just, have a limited bandwidth to develop them in that context. Talk a little bit about how some of your tools and some of what you're doing, and maybe at the Faculty of Pharmacy and the work you do there, you see the ability of some of these new tools to help be sort of reawakening a new generation of these complex natural products that come with these unique glycosylation patterns.
So just the first few words about this big technology we were using in the 90s. So the idea was to make tools, how to measure activity of different proteins binding to lectins, and the digoxin, digoxygenin, was a tag which Boehringer Mannheim developed to label different molecules, because people mostly use biotin, but the problem with biotin, there is endogenous biotin, which is causing trouble. So they made the dig tag, and then I was developing tools with the dig tag. And actually we did develop pretty nice tools for that time, but as you say, this was 30 years ago. Now about these complex bioproducts, I think there is a huge future for that, because for a long time the medicine was ignoring these complex polysaccharides because they were undigestible, and we said, you take them in, you're not digesting them, they go out, they don't do anything. But this was the period when we were actually ignoring the microbiome. And now we know that our life depends on our microbes. And I think the not majority, but a large part of health problems people have in the Western world, particularly in US, are caused by the ultra-processed food. Because ultra-processed food is something which you can put on a shelf of a supermarket and come and buy it a year later and eat it. So if there is a food standing on a shelf for a year and bacteria don't eat it, meaning there is something which kills bacteria in there. So we put all different preservatives to kill bacteria. The problem is when we eat that food, we also eat these chemicals which kill bacteria. And then we kill our own bacteria. So our microbiome gets reduced to the small number of species which can survive all these preservatives. And the other thing is, this ultra-processed food has a very limited number of ingredients. No, just the basic things, some starch, some fructose syrup, or whatever. While we need diversity in our microbiome to protect us from foreign bacteria, because the biggest threat which can happen to us is some bad bacteria coming into our digestive tract and start multiplying. And the first line of defense from that is our own bacteria, because our bacteria do not want competitors to come and take their food. So they fight. But we have no ideas which kind of foreign bacteria will come in. And what is the common knowledge? If you go in India and you drink any water from anywhere, you get sick. But the local people, they don't get sick because they have their own microbiomes which protect them from bugs in that food or drink. And we don't. And the idea of having diversity in the food is that this diversity in the food we eat is promoting diversity in bacteria in our digestive tract. So, and this is where all these strange products, which were never formally accepted in medicine because we had no ideas how they work, come into play. Because if you have good complex polysaccharides, which bacteria can eat, you will cultivate some species of bacteria. And then you have another one for another species, and then you have a healthy, diverse microbiome. And then some bad bugs comes in, and they just got eliminated by your own bacteria. So I think there is a huge potential in these dietary supplements based on different polysaccharide material, which has not been properly explored, and actually it was treated very negatively, even by the glycoscience community.
Yeah.
So we as a glyco field were against all these different glycan products, because the idea was, you can't digest them, they will not do any good to you. This was wrong.
Right. Yeah, it's been a fascinating history, and I really, it's just refreshing to see you at the epicenter of all this now, over the the last couple of decades, as these tools have evolved and sort of leading the glycoscience space forward. So I mean, this has been really enlightening, and it's just been a pleasure listening to this journey, and wishing you really the best with all of it as you continue to move it forward. For everybody, again, that's going to be listening to this show across our various podcast networks or watching on the YouTube channel. Again, you've been listening to Dr. Gordan Lauc, Professor of Biochemistry and Molecular Biology at the University of Zagreb, Faculty of Pharmacy and Biochemistry, founder of Genos, Chief Science Officer of GlycanAge, and co-founder of the Human Glycome Project. Gordan, I want to thank you for taking the time out of your schedule to come talk to us for a little while about all these topics. Obviously, thank you for everything you do to move glycoscience forward in this way. And as we say on our show, thanks for helping to create a better tomorrow via what you're doing. It's just really very impressive.
So thank you for the invitation. I have to say I'm deeply impressed with how well prepared you are and how all papers you read. So
they were impressive papers.
Again, you think it was interesting.
Absolutely. Well, we'll definitely do a follow-up. It was really great to see you.
Okay.